לא מוכנים פה.
כן, איזה...
גם בחוץ ונשנשים, אני לא יודע מה קורה פה. זה בסדר. מסך.
בצד של החולים.
I got to say, be up there.
afternoon, everyone. Thanks for coming this afternoon. It's my great pleasure to introduce Professor David Kleinfeld from UCSD. Professor Kleinfeld is a distinguished neuroscientist whose pioneering research has significantly advanced our understanding of neural circuitry and brain function. His innovative work spans several areas, including the dynamics of blood flow in the brain, the neural basis of sensory perception, and the development of novel optic and computation tools for neuroscience. He is truly an interdisciplinary scientist, combining physics, engineering, and biology, and really exemplifies the type of research we strive for here at ELSC. And on a more personal note, David is a true friend of ELSC for many, many years. He's been on our international steering committee and helps ELSC grow from its early days behind the scenes. So that's one thing. But I just want to mention one thing before I do the Heller lecture ceremony, that in October, we got many emails from colleagues and friends from abroad. One of them was from David, who wanted to help, but he wanted to proactively help and come over. So I just want to thank you. It's not taken for granted in these complicated times. So I really want to thank you for that. And David has been giving many lectures throughout this week. This week we made them work very hard, interacting especially with the students. And as a final bit, he's giving the Heller lecture. For those of you who don't know, the Heller lecture is a lecture series that was inaugurated by Michael and Morven Heller from the UK. And Michael passed away a few years, but Morven decided to continue the tradition and continue and support the Heller lecture invitees to Israel. So I think it's the second time we give the Heller lecture. We gave it many years ago. So first of all, please welcome David Kleinfeld. And David, I would like to award you this certificate of recognition. He can laugh, you can laugh. And appreciation for your contribution to the academic inquiry and exchange at the Edmond and Lily Safa Center for Brain Sciences at the Hebrew University. And Eli, who asked me to swap for him, is signed, as well as Jonathan as the head of the PhD program. So thank you very much.
Okay. Okay, very, very sweet of you. So it's been a nice, it's a complicated time, but it's very happy I came. I learned a lot. David was non-stop. But as I explained, it was the first time I ever tried to explain math in front of a real mathematician. So it's like, you know, it's like playing sports in college. You think you're good, but maybe not after. Okay, so over the last, many years, we've been interested in this problem that's called active sensing, the computation of active sensing. So all the sensation is active. We're moving our eyes, we move our body. Everything is active. We move our hands across the surface, we perk up our ears. If you're a rodent, it sweeps its whiskers back and forth. It scans. Even our eyes are a scanning system for the most part. I mean, this idea of fixating on a point and studying the vision system, maybe something one should think about. And that poses computational problems, and it poses the issue of how you deal with self-motion, and it poses the interesting question of what the motor control is for the sensation, and then how maybe the motor system could actually act as a synchronizing input, because we really want to learn something about the world. The world has certain statistics, certain features, and all we care about is about learning about it. It doesn't have to be pure vision, pure auditory, pure touch. So to maybe it's possible that aspects of the drive for the self-motion can actually act to synchronize all these different sensory modalities. That was our starting point. So let me give you sort of the bottom line in case you fall asleep, or I fall asleep in the middle of the talk. Okay, so the bottom line, this was work with my colleague Martin Deschênes, who recently retired, more recently Fan Wang at MIT, and theory work with, published theory work with David Golan at Beersheba, is there's a common clock, and at least in the rodent, that clock is breathing. Okay, so we normally think of breathing as just something that's there for metabolic needs. But the inspiration clock, this region of the brain that was described first in detail by Jack Feldman, okay, it's not just moving the phrenic nerve and allowing you to pass air, but it's actually sending a signal, projecting a signal across the brainstem from bottom to top in the brainstem, and this acts as a synchronizing mechanism for all different actions, for whisking, for licking, for head turning. And I'll go through various aspects of it. So we have, we don't have a whole picture, or I guess we retire, but we have some solid pieces of this picture, and then I'll end with sort of work in progress, which is getting at how this motor signal actually impacts on a sensory signal, and how we can try to decode it. And this is work that David Hansel is beginning to contribute to as well. Okay, so I think at least for all the students and postdocs here, you've heard the joke by now. You train and you train and you train and you train, and you learn to do great experiments and build equipment and design electronics and write programs and do all kinds of stuff, and then you become a faculty member and you write grants and you teach and you deal with the chancellor at the university. So the work, of course, is done by the kids in the laboratory. So I'll introduce who did what, but I just sort of, no, where's the colors? Oh well, we lost some colors. I'll introduce work by Panton Yao and Ray Liu, and I mentioned David Goldman, David Hansel as senior collaborators. That's not yet published, but otherwise we'll go along. Okay, so I just remind you that these are not new ideas. I mean, this was first talked about by Van Holst in '54, quite a while ago, that to understand sensation without consideration of the effects produced on the sensory receptors by motor impulse is something you cannot hope to understand. Every time you move your hand, okay, you're stretching your ligaments. I mean, things that people describe as proprioception, in fact, are telling you something about how much the hand stretches. The late Steve Schau showed that actually tells you how big the glass is. Wine glass, juice glass, this because of the proprioceptive receptors. Okay, so I learned last night that PDF no longer likes movies. So let's try this. This is a Canadian mouse. It means it's friendly. What's it doing? What's it doing? Okay, so it's whisking. It's fast. It's fast and it's sharp in frequency space. So one aspect of the motion is some frequency that's relatively narrow band. And then it's kind of moving its head around. And, there's jerky movements, it's slow movements. So there's sort of some broadband aspect to the motion, and there's some rapid, sharp aspect to the motion. So, sorry? So I think I could illustrate it this way. So As best we could tell, whether we're looking at movement of the tongue or rotation of the head or movement of the whiskers, the vibrissa, there's some rhythmic movement, and which is sharp in frequency space, and then there's everything else. So this gives us a signature in the laboratory, right? Looking at the behavior gives us a signature. So at least, broadband, you don't know what to look for, but rhythmic, you know what to look for in the laboratory. Okay, so the first thing we want to look for in the sense of rhythmic control is how the animal searches. I think I'm going to stay here because I'm going to fall off the ledge if I keep trying to read the slide. Okay, so we want to know which global variables actually modulate movement, and if we want to understand how an animal can search within its immediate space, it can move its whiskers back and forth, and every time it does that, it spans a region that's given by one of these these pies. But when it sweeps its head, it's sweeping through the largest region at space. So let's now focus on the behavior that allows it to probe the largest region in space. Okay, so this was an experiment. This was done by Sungmao Lau, who's a grad student in the lab. Sungmao somehow joined the world of AI, got paid more money than certainly any faculty member at UCSD does, and then decided he needs to go back to the laboratory. So we're back, and the experiment is to measure motion of the head, which is just due to a gyroscope mounted on a MEMS gyroscope mounted on the animal. And what we're doing is we're having the animal search throughout this large arena, sort of meter arena, and the animal will drop in food at random places. The animal basically covers the entire space, and we're measuring all the angles of the head, pitch, yaw. Okay, we could actually just accentuate the movement by taking the derivative and plotting the velocity, and at the same time, we're measuring breathing. And I think what you notice is there's some sort of high-speed movement of the head as the animal sweeps its back and forth. It's actually sort of modulating the position of its head. So first, let's see, in fact, if behavior has some effect on something fundamental like the breathing of the animal. So what we find is that the animal will search on the ground, like it's performing chemotaxis. And then looking for this food pellet, and every so often it gets lost, and it raises its head and it starts to cast from side to side, a little bit like insects. Our colleague Massimo Vergasolo would call this infotaxis, looking for little puffs of information. So we know when it's in this head down foraging because we get one peak here, and we know when it raises itself up. We just look at the pitch angle, and there's another peak here. We're just going to ignore what's in the middle. The side-to-side movement couldn't care less as to whether the animal is above or below. But what we find is very interesting is that when the animal changes, its behavior, when it goes from sort of searching this odor trail for the food versus losing it and looking up, it actually shifts the frequency of breathing. And it's pretty dramatic. So there's something about search behavior and breathing which are coordinated. And let's see what else happens. So we could look. So what I want to show here is just focus on one thing and focusing on the pitch, of head movement relative to breathing. Okay, so this is effectively the autocorrelation of breathing. Just breathing locked to itself. It starts up just out of the beginning of the inspiration cycle. That's shown here. That's shown here. This is for foraging, and we've measured all these parameters of the animal, but we could focus on pitch. Okay, what we see is that the pitch of the animal has one phase when the animal has its head down, and has a completely different phase, shifts by nearly, not quite pi. When the breathing rate changes, in fact, slows down. And you could quantify this by just looking at the change in the phase of breathing. It's almost pi. So if you sat in the class this morning and you're thinking, how do things change by pi, right? There's some change in frequency, and some change in behavior. So this is just happening. It happens like that. So this is happening due to the dynamics. This isn't some modulator, some complicated biology thing. So then you want to make sure that you say, well, maybe this is secondary to movement of the chest muscles. So you actually want to record in the individual neck muscles. So there's a lot of neck muscles, and it's complicated, but, okay, somehow did a great job. So, I mean, the names of the muscles aren't very important. Some of them cause you to rotate out, and some of them cause you to rotate in. This is the splenius muscle. This is what sort of aches if you sit up in bed with a pillow too long. Okay, we'll just leave it at that. But this is enough muscle to get control. And the point is that we are now modulating, okay, the phase of individual muscles as we change behavior. So this is foraging, and this is rearing. In some cases, this is very dramatic. This is the biventa cervis. I'll just show you. I mean, you can see it's peaking here, and then it's broad peak, and it's moving over to here. So you get these large changes just with a shift in breathing. So somehow, surprising, right? The animal's turning its head, it's modulating it, locked to breathing, and the timing relationship is changing. And basically, when the animal's on the ground, okay, what's happening is it's basically sniffing, it's breathing in as it's turning from out, in. And when the animal is up, looking around, it's breathing in as it's going from in, out. And it doesn't matter which side. It's symmetric with respect to the two sides. So it's having this ethological change, okay, in how the animal perceives. And what paper would be, you know, this is the modern way. You send a paper to an in, you know, figure 7, figure 8 has to be a model, or it doesn't get accepted today. But I think the important point, so this is as a hypothesis, which now drives experiments. You could think that there's an oscillator for the neck. Okay, so this is work in progress. We have located presynaptic neurons, but we don't have this oscillator yet. I'll get back to premotor oscillators in a moment. But the idea is that if you have an intrinsic oscillator and have the breathing oscillator, and the breathing oscillator, we know drives this in a one-way direction from the anatomy, that changing the frequency of one oscillator, in principle, is enough to change the phase relationship. So this is our working hypothesis, okay? And I'll get back to another example where this thing is on solid ground. But this is the idea, that the breathing oscillator and every motor action that your brainstem drives has its own oscillator, and then the rodent, it gets input, okay, from the breathing oscillator. That can entrain it. And the phase relationship depends on details of the frequency. Okay, so what we learned is that the dynamics of rhythmic head turning and sniffing are phase locked in a manner that depends on behavioral state. Okay, so let's look at a broader role of sniffing. And this was suggested already by Wally Welker, 1964, which was the year that the Beatles were first on Sullivan, by the way. Okay, some of your parents might remember. Okay. So we look at many things. Right? We have respiration. We look at licking. Okay, very important. The animal is constantly opening the jaw, licking, tongue goes out. Licking in a rodent is not just to imbibe liquid and food, but licking in a rodent is a way to touch the ground and actually scan the environment in front of the animal. Okay, we look at whisking, which we'll get back to in a little more detail. We look at the nose twitching, moving from right to left. So the nostrils are picking in air, they're coming from the side, so it's really sort of sweeping how it probes olfactory space by pulling in air from the two sides. I mean, head bobbing, we spend some time. A long list of muscles. Anybody could remember all the anatomy, I'm very impressed. Okay. But the point is that these, as you can see, are all locked to respiration. So every time we've looked hard, And these are EMG measurements or behavior measurements. It's always locked to whiskey. Okay, so I want to make a pitch about EMG measurements for the experimentalists. It's fashionable today to have many cameras looking at your animal and get some, 27-layer inception-inspired neural network to figure out what's going on. Sorry, what's wrong with that? What do you lose by not measuring EMG? Anybody have an idea? Okay, if you want to figure out, so you have to start with, before you ever get into the laboratory to do physiology or anatomy, you have to get the behavior down cold, right? And then you start to do physiology, then you start to do anatomy. But if you want to figure out the circuit, okay, you can start at one end or the other and use anterograde traces or use retrograde traces. And to work out motor control, you can use retrograde traces. I mean, most of these are based on variants of rabies and variants of AEVs that go retrograde. So if you know what the muscles are, you can start from the muscle and go backwards and have control. But if you don't know which muscles are involved, you're not going to have any hope to do the anatomy, going back, finding, not the motor neurons, but the premotor neurons, which are potentially the oscillator cells, and maybe even one step before that, the pre-motor neurons. And some of that will take you into layer 5 and cortex. I just want to emphasize the importance of taking the time to do EMG. And it's easier today. Okay, Sam Sobel has these beautiful EMG electrodes. Okay, so what we've learned is that orofacial motor actions are locked to sniffing. It's like inspiration is like a clock that binds the actions together, perhaps binds the perceptions. And let's look at the underlying circuit. So we want to solve, at least, ideally, we'd like to solve many circuits just to get some sense of how nature solves this. But so far, we've solved one. And this began with work with Jeff Moore, who was a student. He's now professor at USC. And then Martin, I mentioned, at Laval, a phenomenal physiologist, just recently retired. So you measure whisking, and which is in blue, and you measure breathing, which is in red. This is a rat. This is a head-fixed rat. And I mean, they're synchronized. There's nothing here that tells you there's a separate oscillator for whisking. Okay, then we want to turn off breathing. So you could spend half a year making a transgenic mouse that inactivates specific cells in the pre-Bossinger complex R. Following Martin's suggestion, we could give a little bit of ammonia. We could record from the pre-Bossinger complex. It shuts down, and the animal starts whisking like crazy. So this simple experiment, this Martin's experiment, tells you that there has to be a separate oscillator for whisking. And in fact, as time went on, every motor action is driven by its own oscillator that could get in training. Okay, then in order to make progress, you have two rhythms, and you're not going to dissociate things. So there are times when the animal will breathe slowly, that's shown here in red, but it will whisk quickly, that's shown here in blue. And looking at these traces, you can see a couple of things that, like here, that's sort of typical best example, is the animal was just starting to whisk, the breathing cycle started, and it just reset. Okay, the breathing just reset the whisking. So you have two frequencies, one that's resetting the other. So in the laboratory now, this gives you a signature, right? Two different frequencies to look for. Okay, so this work has been, published now for a decade. I just want to give you a little bit of a flavor for how the construction of these experiments. So we do exactly what the NIH tells you not to do. We go on a fishing expedition, but I mean, here, right, Israel, Sea of Galilee, fishing is a big deal. So you start at the pre-Bossinger complex, and you figured, listen, this thing is driving all these other motor circuits, so let's just walk, right? So again, there was this notion that the brainstem is hard to record from because you're starting in the cerebellum and you're going down 8 millimeters, but you basically look, say, for one edge Okay, in our case, we look for the anterior edge of the breathing complex, and we know where we are, and then we walk. And we find places in which we find cells, like shown on the top, that are whose firing rate, which is the black trace, is locked very nice to breathing. You put a spot off to the histologist. We find other cells in which the neural activity, shown here, is locked very nicely to whisking and not at all to breathing, right, which is goes to this long pause. And you get to make a spot, and eventually you find a region that has a number of neurons that are putative cells for this breathing oscillator. Okay, so time goes on, and we get to the point that we can actually go in and you find out where it is, and then you can make intracellular measurements. Martin, invented this technique of juxtacellular recording. She sort of kind of snuggle up to the cell, just kiss it very lightly, record spikes in the cell, and then at the end blow through. And you can make intracellular measurements in some cases, but at least deposit a dye. And what you find is that you get two flavors of cells. You get cells that fire roughly in phase with whisking, and you get cells that fire roughly out the phase with whisking. So there's two populations of cells, and you can see them very nicely. One we call one protraction unit, one retraction unit, for sort of obvious reasons. Now the other thing you can learn, and we make again, as you see later, we're big fans of high tech, but you can learn. The most informative measurement in neuroscience is what can you do to measure from a cell to learn the most? Do fMRI on one cell? What can you? You want to be intracellular. You're inside the cell. You're listening to the cell. You're not just getting the spikes of the cell. You're also getting the currents that are coming into the cell. So we're intracellular in the motor neurons, okay? And what we see, in fact, is we kind of look at this and we see what we thought was an inhibitory current, and then we can inject chloride, so we can go from below the chloride reversal potential, the normal chloride reversal potential, to above the chloride reversal potential, and we can see that we're going from being locked out of phase now to being locked in phase. So we learn that this oscillator actually has an inhibitory output. Okay, so we learn a lot, and we get to a point, and then all of a sudden you have to sort of up your game, and classic physiology runs out. We have a long-running collaboration with Fan Wang at MIT, and now we start to, once you sort of shake out the essential anatomy and physiology, then you can start to find out which gene products are expressed by the cells in the regions of interest, and iterate towards maybe using genes to your advantage to have cells express the label. So one thing that we found is that many of the cells are parvalbumin positive that are part of this. Now it's going to turn out that they're all inhibitory cells, but it's important to remember that in cortex, the inhibitory cells are parvalbumin, but elsewhere in the brain, they're whatever they want to be. Okay? So the idea is that there's a system, it's called the Cre-lock system. Many of you are familiar with this. Cre is a plant enzyme. It'll flip around a piece of DNA. So if Cre gets together with a piece of DNA between lock sites, it can flip it and turn it from nonsense to something that's active. And Fan made animals that, in this case, half the neurons that were the parvalbumin neurons produced half of Cre, and then we had the other half of the Cre come in by putting into a virus in the facial motor nucleus. Right? We knew this was a nucleus that drive the vice versa, and then a miracle happened. You had Cre now was two proteins instead of one protein, but they came together and they functioned as an enzyme. We were able to check this with anatomy, and in a sort of molecularly defined way, we're able to go in and transiently inactivated group of cells and shows that it kills the oscillation. And the other thing I've shown here, as I said before, the input to the motor cells is inhibitory, right? So the only way this could work is if there's some positive input, and then this inhibitory oscillation is coming in on top of it. So you can see very nicely that when you kill the oscillation, the whiskers go all the way forward. And when the oscillation is going, it's kind of like doing a butterfly stroke, right? You're pulling backwards. Well, freestyle, but it's doing it with symmetry. Okay. So this is kind of confirmatory, but once we have this kind of game, we could use these techniques to sort of map the nature of synapses now from the breathing center over to the whisking oscillator, and we're able to use these to actually map between whisking oscillator cells themselves. And okay, so here's an example, which is inhibitory projections. We wanted to look in the pre-Botzinger complex. Okay, so we map from our Vibrissa oscillator, and this is going to go back to the pre-Botzinger complex. And what we find, in fact, is that all the synapses from the breathing oscillator to the Vibrissa oscillator are also inhibitory. So breathing inhibits the Vibrissa oscillator inhibits the motor neurons, inhibition of inhibition. And that's why it looks like breathing and whisking are in phase, right? Because it's two negatives. It's two negatives. Okay, so this is a cartoon. And so at the end of the day, the papers now have been, it's sort of a series of 3 experimental papers over a decade. It's outrageous how long it takes for us on one little circuit, right? We have this picture. There's a breathing oscillator. It inhibits a group of neurons that have inhibition among each other. Now, if the animal is breathing very quickly, the pre-Botzinger complex entrains the system, and you have this feed-forward pathway. And you basically, the pre-Botzinger complex, inhibition, and you're whisking in phase. And it's not, I would say, so terribly interesting. But if you're breathing slowly, there's a group of inhibitory neurons here, and a group of inhibitory neurons here. They're weakly inhibitory on each other, okay? But they're strongly inhibitory between the two populations. And this will make an oscillator. So we can test this. And okay, first of all, we just simulate the whole system. Okay, so simulation science is kind of oxymoron, but we put in as much detail as we know from experiment. And this is a fortunate case where over time we accumulated a fair amount of detail, right? I mean, we don't have a connectome, we have a lot of detail. We know experimentally that you get a kick from breathing, that's red, and then the system just oscillates and winds its way down. And we could calculate the output from all these different cell groups. I don't think that's important, but we look at the calculated behavior, and the flavor of the calculated behavior sort of matches the flavor of what we see in the laboratory. But as I mentioned, we get a little bit of insight because we have this feed-forward pathway when you're breathing very quickly. And again, you're breathing, in this case, 1-to-1. So every time you breathe, you get a whisk. That's this feed-forward pathway. And again, we're simulating this numerically, and it kind of works out. And we can go to the other extreme, where breathing is turned off, and we're just looking at these two populations of cells, and it's a numerical simulation, so it has some noise. And again, we get this very nice pattern of whisking. So I would say from our perspective, this is kind of a self-consistency argument, is that we accumulated a lot of experimental variables. We made up a reasonably simple conductance space model. We run it on a computer. I don't know what David Golem did at night to sort of jimmy the numbers, but it kind of works. But how much insight have we gotten? I would say 0, okay? I would say 0. So, you want to get insight, you hopefully have accumulated enough data, you can make some very brutalized model that seems to capture some features of the experiment. So, okay, we have a group of cells. You have connection strengths among themselves, and we're going to take this kind of averaging approach. Bart Ehrmantraut, Chaim Sampoinsky here, worked on this approach. So we have very few parameters. These cells interact on themselves. That's one term. They interact with their neighbors. That's another term. Okay, that's 2 parameters. There's some outside input. And then let's try then to understand the dynamics of this circuit, because we've accumulated enough data. Now we can go ahead and brutalize it. Okay, so we do this averaging over the conductance-based equations, and we learn a couple of things. So let's look at the red traces. Okay, that's the important thing. The first thing we learn is that this circuit has 3 phases. Okay, so the other thing I should add, which I didn't mention, is that there has to be some slow current, because it's an oscillator. You need two time scales. So there's one parameter, which is this difference between connection strengths. So we learn, first of all, the connections between the populations has to be stronger than the connections within the population. That's critical for it to oscillate. And as we move in this direction, there's a region where the connections are too weak and the 2 populations are independent. Okay? And finally, we get to a point where they can oscillate, right? Where the inhibitory input for one group can beat, okay, the slow kinetics of this potassium channel, this adapting channel, and the system will oscillate. And basically, the amplitude is independent of this parameter, okay, but the frequency is dependent on this parameter. And then if we push it even more and we make the connections even stronger, the system becomes bistable. So bistability between populations like this is sort of described in some of these high-level experiments like binocular rivalry. Okay, but you have one very simple circuit that can exist in all 3 regimes. You never get this from numerical simulations. We have an idea that the amplitude is controlled by connection strengths, and we could play with this because we're capable of going in, and as we did, and affecting gefrin, which is the binding protein for GABA A receptors, and you can start to change this parameter. Okay. On the other hand, the external input affects the amplitude. That is qualitatively under control. And the other prediction is that this interaction strength will affect the frequency. So I think we learned something from this. I think that going ahead and doing this kind of analysis gives us some insight that nature decided to do things in the modular way we learned about in engineering. All right, that you don't change one parameter and the frequency changes and the amplitude changes. External input focuses on the amplitude of whisking, which is what the animal wants to do. The frequency is some internal parameter, in this case, but nature turns out to be modular. And I thought this was a lot of fun that came out of this analysis. Okay, so this was just what I said. So now let me look at this issue of that there's a larger role for these rhythms in sensory coding. Okay, so any questions? Is this more brainstem than you guys have seen in the last, ever since ELSC was formed or something? You went to brainstem.
So is this the fact of having 2 populations, is this common to the other oscillators, to the leaking?
Yeah, the leaking oscillator is work by Jun Takato and Fan's lab. We don't see this yet, okay? I mean, so far, it's not done. It's not done. It doesn't seem it works that way. The head motion oscillator is really at a much more primitive state in our own lab. So I don't know. Okay, I really don't know. I think the one thing that's true is there's very little excitation in the brainstem. Okay, it's rare. It's just rare. I mean, there's no no in biology, but it's just rare. Okay, so I want to move up a little bit to a little bit more familiar territory. So a little bit towards your question. So you know, as a group of Mike Economo, Dan O'Connor, Fen Wang, David Cole, and myself, we're interested in these very many rhythm generators, you know, for chewing, wicking, head turning, whisking, etc. All of which appear to be driven by the whisking oscillator. But we're also interested in feedback, which I'm not going to talk about. But feedback, of course, requires understanding about how these signals get into sensory cortex. So let me give you a little bit of a flavor about how this motor modulation affects the sensory signal. Okay, so what I'm going to do is like ignore everything except this feed-forward pathway, right? And move into a little bit more familiar territory to people, thalamus and cortex. Okay, so what I described so far is, you know, like many children of neuroscience, we started in cortex and then at some point realized we didn't understand anything, so we wanted to understand at least where the motor signals came from. We took a dive down, but now that we know a little bit, we want to come back up and understand something about the pathways that affect the sensory signal. So I'm going to show, this is unpublished work, some of it is reasonably complete and some of it is very incomplete. I just want to make one point. There are two signals that come up in the sensory pathways. There's the self-motion signal, okay? And then there's a touch signal, like an x-afferent signal. And certainly in the vibrissa system, they are both going on the same pathway. So whatever the challenge, there's a challenge here to understand how two signals come in the same pathway and how you get rid of one or use one to your advantage. Maybe the rhythmic signal is telling you where the whiskers are. Okay, so this gets back to old problem. Okay, so for many years, okay, this is now represents close to 15 years. Different people have done experiments on recording signals in the brains of the vibrissa system in rodents, and they all get different answers. And then so the question was what? And it's a long list of papers. I mean, a few people pop up multiple times. My sometimes friends, sometimes competitor, Carell is on here. We're on here. Dan Pillman is on here. But basically, the animal is whisking back and forth. It's whisking an angle, okay? But there's other ways to describe the position, and particularly if you think of the motor control, you can think of where you are in the oscillatory cycle. And that is just plotting the phase, which seems to be sort of a theme of this visit, okay? And so for as long as your signal is rhythmic as a sort of data analyst issue, you could turn the signal into a rapidly varying phase, like I showed at the beginning. We have a rhythm, sharp, and then slowly varying broadband set points, which would be the top position, and amplitude. So let's see what the rodent really uses. So these are experiments by Ray Liu, and there is no reason a priori to assume that it's a change in the nature of the behavior that you're going to use the same coding strategy, which means there could be a big difference between doing an experiment where you're doing broadband exploration, or for the rodent, it's sweeping in front of itself, broadly, looking for something, okay? As opposed to touch a pin, get a treat. Touch the wrong pin, don't get a treat, okay? So we had an animal, which is, I'll show you a movie in a little bit. It's chasing a pin. Okay, this is like one of these incredible findings. You have a head-fixed rodent, which is a little bit like, I don't know if you find Ryanair ever. That's the closest thing I can think of. And the animal is chasing a pin. It's like getting a border collie and having a ball. The border collie will go, I mean, you're exhausted. The border collie will go for 4 hours chasing the ball. The rodent, the mouse, head fixed, just, you know, acclimation to the apparatus is just chasing that pin. I mean, you know, the ICO committee comes in and tells it to stop, right? Okay, but the point is kind of very interesting. As it shifts its, you find that on its own, it's chasing this pin, which is at every possible angle. It breaks into one of two modes of whisking, and one is with the whiskers primarily forward, and it's extremely rhythmic, and one is with the whiskers somewhat back, or to the side, and it's somewhat arrhythmic. You can average all this together. You can see there's a shift in set point. You can see this, if you want to do some, you know, descriptive statistics, there's a shift in frequency, shown here. There's a shift in amplitude. So maybe, okay, maybe when we do our experiments and we're trying to understand something about what's being coded, we have to get the behavior under control. So we wanted to look, and our interest is that we know something about how signals are coded in the thalamic nuclei, the brainstem nuclei. So we really wanted to start to look at what happens, how are things coded. I'm just starting here. This is thalamus. This pathway gets self-motion and touch, and it's going into layer 4. And as an experimental program, right, we want to see what things look like in thalamus. But the way to see what's in thalamus is to see what the axons are doing in layer 4, because they're not so deep. And then the way to study the interaction is, well, layer 4 is maybe 400 or 500 microns down, and that's deep enough that you're beginning to destroy the focus in an optical system. So we borrow an old trick, 1972 trick from the astronomers with adaptive optics. Everybody's seen the Webb telescope with all the panels, you know, the whole world was following the telescope. Okay, so not quite so big, but we do something similar to, we use, Avi mentioned, we play with blood vessels. We use blood vessels as a guide star. We use the guide star to correct the wavefront, and then the wavefront allows us to look at individual spines down at the depth of layer 4. So that allows us to look at presynaptic photons, which is looking at thalamic cells, and then it will also allow us to look at layer 4 cells directly. Okay, good. This is the work of Ray Liu. It's long published, and let's see what's going on. So Kaspar Pogarski now at the Allen made a beautiful sensor of glutamate. I mean, the signal to noise is like 9 or 10. It's outrageous. I mean, it's actually, in my opinion, it is the way to record spikes, because the signal to noise is high. The problem with doing no offense, the problem with doing electrical recording is that you have to be fast or they're gone. But these cells don't spike much faster than 10, maybe 20 hertz, so you can stretch out the pulse. And that's what the sensor does. Okay, I had nothing to do with building a sensor. Most of Kaspar's work was directing it to either presynaptic photons or postsynaptic spines. But I think the important thing is this is almost trivial. I mean, we're just putting this sensor into thalamus, and it just transports up to the boutons. We can measure whisker angle. We can measure the boutons, and these are just some examples of the response. Okay, so let's see what this looks like. And okay, so I said there's two modes of whisking. There's sort of a high phase and a low phase. So what's being coded? Okay, there's always a little bit of a complicated problem, but the one thing is that we were able to record tens of thousands of boutons, and at that point, you could use information theory. So I think in 2 months, right, Tanya Sharpie will come and make you suffer through information theory. So she made me suffer, because we're friends. the point of this analysis is not to say that you could figure out the algorithm that tells you how to decode a signal into, and by coding preferably phase, where I am in the whisk cycle, or angle, right? And obviously some angles all the way forward are also the top of the phase, so they're not uncorrelated quantities. But it gives you a sort of a fundamental measure and an unbiased measure. So what we find is that on average, okay, most boutons are preferentially current, these are for large whisking, are coding phase and not angle. So if I just look, each of these points is a measurement. Okay, this is the neutral information projected onto phase. This is projected onto angle. And these are just a few examples where the coding, say, for A is where phase, whereas A is here. You're coding phase better than you're coding angle. Okay, and vice versa. Okay, so just you take it as you will. But if we take these numbers seriously, then we find, I think this is something unexpected, that when the animal is in this high set point, very rhythmic whisking, that there are many more neurons fractionally that are coding phase over angle. Okay? And then when you go to this low whisking, the proportion flips. I mean, the number which you're coding well drops, and the proportion flips. So the general lesson here is that the nature of the behavior, okay, this is the nature of the motor act, okay? Set point change, regularity change will have a tremendous impact on the nature of the code. So you can't just say, whatever, I'm measuring arm reaching or whisking or something and change the condition, okay? And if you look at what the coding is, this is about 85. Okay, this is only about a third of the boutons that has some significant mutual information, which means we don't understand 2/3 of what's going on, but, you know, plan to be in science for a long time, so it gives me something to do. So, you know, there's about 25,000 boutons. This is about a third of them that from one animal that we were able to measure, and you can see that all phases in the whisk cycle are coded. Okay, and if we try to now, if we replot this for these low amplitude whisks, it's essentially flat. Okay, so how do we look at this? Ah, I get to show another movie. Okay, come on, it's going to work. Okay, so look, this is what we had. This is bouton number, and this is phase. And bouton numbers are coding phase. So in a sense, as you go around in a circle, this is one sweep. And then as you go from neuron to neuron, that's also 2 pi. And we thought this is just a data coding method, but we thought it's a nice picture of what's happening. I mean, what physics talk is today without a torus? Okay, so there's a group of boutons that are active, and as this bump goes around, it's just activity passing through all 8,500 boutons every cycle. So all parts of the brain, as we'll see in a moment, have knowledge of where the whiskers are, where the vibrissae are in space. Okay. So back here. Okay, so sort of as a side issue, it turns out that there's actually some spatial coding for phase. This doesn't. So each. This looks like random M&Ms, but each dot is an individual bouton, and each color represents the phase. So I mean, there's lots of ways. So there's a thickness here, which is 100-something microns, and then this is the size of the barrel of the cortical column, and I'm just candy coloring phase. So what you can see is not just that the system codes in phase in these big whisks, but it actually has some organizational structure. Okay, so this is the animal. It hasn't even touched anything yet. It has a very active, vibrant input. So what happens with touch, right? So we're going to walk our way now into the thalamocortical system. All right, so to address that, I'll show you a movie in a moment. These are two snapshots from the animal. This is the mouse. It's chasing a hare. There's a camera. And you can it's high-speed videography. As I showed you before, we measure the angle. We just decompose this into phase, Hilbert transform. The animal whacks into something. When it whacks into it, you can measure the curvature. Now, 1 over the curvature gives you the torque, or as the engineers say, the moment. Okay, the moment. And now the whisker, it's a little uglier because it's a tapered beam. So you actually have to do this numerically because you have to know where you touch and you have to know what the taper is. But we can go from curvature and contact point to bending moment. So just the last movie, please. Come. Here we go. We're just moving everywhere, and they're chasing. It's really like a dog at a ball, not a corgi, but like a border collie. We had a corgi. Corgi was a queen. I mean, the queen picked well. Okay. All right. Let's forget about this. Let's get back to real science. Is there a question? Okay. Did that. All right. So one of the things we learned is that this self-motion signal, it hasn't touched anything, right? This was the signal that was generated just by moving, just by some inertial effect of having the whisker inside the pad is causing some compression. And if the animal touches, you could ask, does it actually excite at the same phase? Does this self-moving signal act as a reference? Okay? So this is a free whisking signal, and then we have the animal touching. This is just an example of the touch signal. Okay, again, this is recording from glutamate release from boutons. And if we make a plot of the preferred phase where you get the biggest signal at touch, and against that, we plot the preferred phase for free whisking. I would say, given biology, it's about as good as you get for a straight line. Okay, so the other thing you could ask is this. There's a lot of parameters going on. I mean, this is we're coding phase, but it also depends whether the animal hits close to the phase, far from the phase, that the torque is going to vary, that it hits with, okay? So there's a notion of invariant, and I think a lot of us are used to this from the visual system, because you could say that there are neurons in V1 that respond to the orientation of a bar. But if you change the contrast, right, that tuning curve is the same. It's the same width. It's just rescaled. But you say it's an invariant, right? You don't care about the width. The same thing may hold for spatial frequency. So it's very interesting to find, okay, that I think this is the most dramatic data. We're hitting at the same phase, same angle here even, and at all different distances from the phase, right? Torque, R cross F, right? You learned this in high school, I hope. And so here's all different torques, but the response is essentially the same. So the animal is throwing away information. All it really cares about is this angular location. It doesn't care how hard it hits, okay? And you can find actually pieces of this in the previous literature. So what have we learned? That the thalamocortical boutons encode touch, and they close it in terms of phase in the whisk cycle. So this is telling us that a signal that goes all the way back to the breathing clock, right? Which is in training the whisking clock when the animal is actively searching. It's making its way into setting a coding structure into cortex. That's the deep issue here. And also you find out that this signal is invariant. Okay, so that's kind of it for what we have on steady ground, but I thought I'd give a little bit of a flavor where it's going. Many years' work from Panton and Ray. So you want to know what's coming out of S1. And so we started here already, like 15 years ago, and we did this with electrodes. So we had very few samples, and again, we found out there's a little bit of modulation with rate. I mean, that layer 4 cells are pretty quiet, maybe 10 times quieter, less spikes than when you touch an object. And the result we saw in thalamus, that if there's a preferred phase, okay, for whisking in air, this is a log scale. It's a huge impact. Okay, this is where the cell is most responsive to touch. But we had no idea how to make further progress because we didn't have molecular tools or anything. So I'll just sort of close with sort of what our ideas are going forward. So from the past, with a sample that had to be biased because we were recording, we didn't know where to stop our electrodes, right? We couldn't, because you had to have some spikes. And this is what my colleague Harvey Carton likes to say is, if you want to really do it, you need to take inventory. So I think one of the beauties of doing things with optics and not with probes, and with due respect to my good friend Tim Harris, not with even neuropixel probes, is you want to see all the cells and ask what the cell is doing. Like we learned that only 1/3 of the cells are interested, and maybe 1/4 are interested in phase. So we know we're missing a lot. So here we're using, like we're using voltage-sensitive dyes. This is ASAP3 from Mike Lin. He's sort of part of the family. He was one of Roger Chen's postdocs. And what we're recording, the nice thing now is we're recording the transmembrane potential in layer 4 cells. There's an animal, SC, NN1A, whatever, it doesn't matter, that we could only put dye in those animals, and we could record again whisker angle, whisker phase, the dye response. It's an imperfect world, and this gives a preferred phase, but these are all sub-threshold potentials. So it's a funny system. So you have a very strong thalamic input, and the cells are not firing. So this sort of leads to, I'm going to leave you with a hypothesis, which is sort of out of the air going forward. What are you getting from layer 4? Thalamus is already telling you which guy is most active, right? So I just call it the radar hypothesis. You could call it the alarm clock hypothesis. Call it whatever you want. You want something that it's quiet until an event happens. So you want to get detection. And then once the event happens, you want to say this is where it occurred. So you have this thalamic input that's coming in, rhythmic input. Comes here, comes here, comes here, just like I showed. Different boutons, sets of boutons have different preferred phases. They come into these excitatory cells. Thalamic inputs come into inhibitory cells. Add stronger, stronger. Okay? I'm not showing it, but voltage dye measurements in the inhibitory cells shows that they're active. So we're not intracellular yet in the excitatory cells, but it looks like these cells have this competition. They're being inhibited, they're being excited, and they're quiet. That's a running hypothesis. So the system is then very quiet. But when you hit something, okay, when the animal touches something, you get this barrage of spikes that comes up the trigeminal to thalamocortical pathway. And so that's the idea. In fact, we started now to measure. So again, it's an incomplete story, but just the flavor is that now if there's spikes, listen, optically, nothing beats a calcium dye, okay, with all its problems. So this is GCAM 8, next year it'll be GCAM 9, but when you guys grow up, it'll be GCAM 20. But I think the point is that you get very, I mean, this is 800 units, right? You get very sharp phase responses. If you plot it, you get the very sharp response. So in the thalamic end, we are pretty solid as to what the nature of the coding is, and we're just exploring this issue at the cortical end, that it's acting as kind of threshold gate. And then all of a sudden it tells you where something happened, and the same kind of invariance occurs, not surprisingly, in cortex. I could show this later if people are interested. Okay, so just to end, I think the idea is that inspiration, I think the big idea, I would argue, is that the breathing clock, okay, in rodents, is a motor-generated clock, and this is something that behaviorally binds all the senses together. And I talked about how this coordinates orofacial behaviors and head movements. They're sort of locked to this oscillator. The locking depends on the nature of the behavior, and then of course the nature of the sensory signals themselves will depend on the nature of the behavior. And we would argue that this self-motion is an essential part of the sensory system, and if things work out, I mean, basically this threshold operation in cortex is cortex using its own, brainstem generated rhythm to its advantage to tell it where an event took place. And okay, so thank you, and we'll see where this is going. Questions?
Thank you, David. Can you generalize it to bigger animal with much slower respiration rate?
Like a big mouse?
You.
Me?
Yeah. You take 15
The whisper system.
Okay, I swim, I don't...
per minute.
Yeah, okay, I swim, I don't whisk, but you might say they're very similar. I mean, although my back can't take the butterfly that much these days. Okay, this is a problem because, I mean, we don't breathe slow enough to walk to these things. But so I think there's something, there's two things. So, okay, I can't have a direct path, right? So maybe that's a weakness. Okay, but two things are true. Your eyes, I think as you know better than I, are darting around with a sort of typical time scale of around 200 milliseconds or so. It's as though you're picking, you have a random process, I don't know with what statistic, and you're just picking it out of a bin. And you're jumping from location to location, like you're making saccade. So that's the first thing. So that, no one's really looked about how that time locks to other things like hearing and olfaction. Okay, so I think this has to be looked at in people. Now, there's no reason it has to be a rhythm. I mean, you're just saying there's some underlying time scale. So it just could be in the rodent that it's fast enough compared to the natural motor rhythms that that's what nature picked up. I don't know what, sort of random number generator with an average frequency of 5 hertz is sitting in your brainstem. But I think it's, I would argue it's something to look for. Okay, I would seriously argue that it's something to look for. Okay, the other thing is that in the last literally few years, right? How do I know this? Because Stan and Dan and Mike and David Goldman and I wrote our grant renewal, right? So you have to, everybody's a genius. There's growing large literature on people looking at these breathing signals and in general the fast motion of wicking. Some nice papers in the colliculus. Dan sees this in motor cortex. We saw this long ago in motor cortex. So all these fast signals are permeating throughout cortex, and they're just popping up, including breathing signals. So they're popping up throughout the rodent literature. That's not answering the question about big animals, but it seems that people coming from very different directions, like totally high-level cognitive mouse physiology, are seeing these fast signals. So maybe there's something to it, but hopefully connected. So I don't know the human thing. I mean, it'd be nice to explore it, right?
It would be nice, but it is just too slow for me. Because as you say, usually our motor rhythm is about 2 to 3 hertz. Eye movement and so on.
No, yeah, breathing, I'm sure, I'm not sure. It's probably not it, but I mean, it'd be nice to know if there's some signal that's common to multiple, that at least when you're interested in something, that the sniff rate and maybe the eye rate and moving of the ears
becomes common, at least for a limited epoch in time, right? At least for a couple of sniffs, becomes you get a synchronized timing across modalities. I mean, that's the only thing that matters. It's a synchronization in time. Yeah.
Two things I was surprised you didn't raise. One is that the hippocampal formation
Yeah.
produces an EEG signal,
Yeah.
theta, which is really clockwise, and at this sniff cycle, at the sniff rate. And the question is really, is it the cortex that's driving the brainstem or the brainstem that's driving the cortex? And the second issue is that I'm not sure that I buy this sniffing as being breathing. Sniffing, I think, is much faster in rats and mice than breathing itself. But what sniffing does is it brings pulses of air with smells into the olfactory system.
Yeah.
And that's phase locked with the same sniffing and vibrissae movement. And so the olfactory system is getting pulses at 8 hertz or 7 hertz or something like that, the theta phase. And the olfactory cells, the receptor cells, are then driven at this rate. What's more than that, they're phase locked to the sniffing cycle, and different odors are phase locked at different parts of the phase.
Okay, I want to argue with you about that.
Okay. So that claim was made in the. We have a paper on this. That doesn't mean it's right.
That just means we fooled maybe 3 people and an editor. Okay. But the theta rhythm and the whisking rhythm and the breathing rhythm, at least the breathing rhythm when the animal is excited, are pretty close. I just wanted to pull up a paper. If you just bear with me for a second. Yeah, okay. Yeah, here it is. So we looked very carefully. Matt Diamond looked then later, and I would argue the following. We recorded, Runeberg is a physiologist now at the University of Copenhagen. Some people know him here because he did this business of looking at opposing muscles as part of a single rhythm. He was a graduate student in the laboratory. We measured from about 20 animals over the course of his career, in which we had electrodes in hippocampus and as a reference and as control, and we also had electrodes in the S1 cortex for different whisking tasks. And if we looked over long periods of time, which means long compared to the autocorrelation time of both signals, then the hippocampal theta rhythm and the whisking, which we now probably interpret as breathing, fast breathing, okay, sniffing, were uncorrelated. Now, Matt Diamond had a paper some years ago. Actually, Naka Mionoski and I wrote like a little news and views, and the rat would come up to a choice point, and it had to make a decision. And within the kind of 1 second, so we're talking like 5, 7 cycles of the decision making, you get synchrony. And then the synchrony would leave.
Did you say that the somatosensory was recorded from S1 rather than from the vibrissae itself?
I mean, we recorded from both.
What distance between the two?
No, but wait. Okay, so, I mean, in those experiments, we recorded EMG, and we also recorded from S1. And they're locked. They're locked. I mean, I believe this very strongly. It's been, I mean, otherwise, well, it could be, but I think, 20 years of stuff goes up in the bonfire. I think they're strongly locked, but just the theta rhythm and the let's just say breathing rhythm, or is only locked when the animal is very is at a decision point. Now maybe there's a deeper issue, but I don't think anything's been done. Okay, the other thing about I just want to say about breathing, I maybe it's wrong. My understanding from the breathing literature, a lot of it's Nina Ramirez, Jack Feldman, of course, is basically that you get something like this. This is just a histogram breathing. And when the animal is sitting there sort of waiting, it's breathing down around here, 3 hertz, 4 hertz. And then you just get this transition to sniffing. And there's one pattern generator. And that pattern generator, if you record from the breathing complex, okay, that doesn't mean there aren't 2 circuits within the pre-Botzinger complex. But if you record from the pre-Botzinger complex, you get a continuum. It just sweeps from low to high. And this is the distribution we get. And we, I think, you know, this is already over a decade old, but, you know, Jun Takado, others, Vincent Provosto and Fan's Lab, have pretty much reproduced this in their work. So I think it's one pattern generator, and it's kind of an amazing pattern generator, the breathing generator, in that it has a very large dynamic range.
But 8 hertz are little tiny pulses. I wonder if any oxygen actually gets into the lungs from the sniff. But maybe we're too much on this. It's time to...
Okay.
Okay, we're already a little bit late, so let's thank David for...
Think about
dynamics is connected to brain dynamics, connected to field. Things you can do with field. This link is weaker. So the link to actual measurement is weaker. I also talk about spikes and about rates. Second thing is Jonathan had wanted to cancel the meeting because his flight was delayed. He's just landed.
Will he be here or I can wait?
I don't know. That's what Sal called me.
Because, yeah, because I was originally going to go to the driver told me that they're closing down the roads.
Yeah.
And then it's
How much my
so I'm going to hang here later. Maybe if he can make it, then I'll wait.
Okay.
Okay, just ask. Oh, yeah, he's tired from the flight. So maybe it's an excuse. We did meet before, I know Anna. Well, yeah, good ground together.
Well, that's my Anna's.
No,
She's doing well. I think she's doing better. I mean, I think she's more, and then she's better at the science. Oh, I love it to do it. I don't know why that helps. Why that helps you something that's very beyond yeah, but you know, we have like a group with many trainees and me. So, okay, yeah.
So say hello.
I will.
I'm in touch with her all the time.
I think
First of all, very, very cool talk. I mean, I've read a lot of papers before, but it was very cool to see. And I have a couple questions. Do you think there's any feedback on the pre-Botzinger breathing generator by any of the premotor oscillators? Because I was thinking that when you showed that the phase depends on whether the animal is rearing or not, like maybe there's some rearing oscillator that's a breathing oscillator, and then that's sort of influencing.
Well, you know, one point that was, okay, there were some critiques that were raised that we couldn't address. Okay, one is when the animal rears up, I might be changing the mechanics on the chest. When it's down, when its head is down, its chest is falling. And so the pressure on the chest is less than when the animal is riding. Okay, so that could be, this whole thing could be generated by sort of feedback for the muscles onto the nephrotic motor system. So we couldn't really do a proper, we thought maybe we could look as a function of angle, what the frequency was, and we didn't see anything. We thought part way, but then, it's like, so it's not like you have a lot of cycles to average over, but you sort of hope that maybe you catch something. Yeah, so I don't know.
Where do the pre-bots get inputs from?
Well, it gets a lot of feedback. I mean, it gets input from, right below the preboscopal complex is this place called rostral ventrolateral medulla. And RVOM is sensing oxygen concentration within the bloodstream. And it is, that's one feedback. So it's very sensitive to oxygen and pH, which is really sensitive. I don't really have a good, a lot of this is known. I just don't have, I'm missing a lot of basic physiology. Muscles in the diaphragm and muscles in the chest. It's very rare. You go running. You synchronize. You sprint. You really synchronize. Maybe every other step or something that you become very synchronized to movement. And I think it's really because of compression. So I don't know how this works. Compression is known, but not known to me. Yeah. So it's tough. I mean, I'm not sure how important it is, but it's sort of telling me that your sensory is very influenced by your motor act. I don't know how much of this has made its way into sports. I have a kind of interesting physical therapist. Is that destroyed my back early on. Yeah. But he's kind of sensitive to this breathing sports thing. So,
I spoke with Jonathan and he won't be able to make it.
Okay.
You're not going to Tel Aviv now?
No, I was told that there is the main road is going to be closed, or is closed, because they're repairing, they're making a bridge or something.
Yeah.
And I was
It's
warned.
a story.
What?
Yeah, it's because they
Well,
don't want to do it on Shabbat, so they're closing it on the
Okay, so I was warned that to
Yes.
go and then to try to get to the airport is going to be dangerous. So I'll call my friend and just say it's off. And I'll just go to the hotel, try not to fall asleep. Yeah. It's not Shabbat, but it's tomorrow.
Yeah.
I know that
that's the
much.
point.
At least
That's
that
the
much.
point. No,
That
no,
much
that's the
religion
point.
I know.
My clock is okay.
Okay.
Okay,
You want to hang out? I told the driver to pick me up at 6 because I thought I'd take the drive.
6.
Tonight. I'm fine. Don't worry. Pick one.
So he's taking you at 6 to the airport?
To the hotel.
To the hotel.
And then I want to I thought I'd shower and take my jacket off. Dress like a slob and go on the airplane. What are you studying?
I'm sorry. I started the PhD program in first year.
Yeah.
And I also, I work with David and
TD? David Owen?
No, with David.
Oh, you?
Stock, yeah.
But now our project is we're trying to separate motor with different orofacial motor behavior.
The first thing when Mohammed got in the lab that I had to do is read your papers actually. Because I totally, the thing is, I was I really think the key to understanding the basal ganglion in the striatum is to figure out how there are changes that are occurring onto specifically the cells, whose weights you want to change, and whose weights do you want to change, the cells that just did the action that you want to increase. And so how do you do that? I totally agree with your philosophy. Whether it's cortex or basal ganglia, start as close as you can to the action, the output, the muscle, and go with
Come to my office?
Yeah, wait, I gotta remember, which side, this side or the other side?
You can tell.
It's this side.
This side.
Yeah, but you go to the, there is stairs, first floor,
Yeah,
okay? That would be the first to the right.
okay,
Okay?
I'll find it.
2 minutes ago.
Yeah, so I totally really love the idea. The lab has invested a lot in this, putting cameras and training a network to figuring out what the best behavior is. So I'm like, I'm curious actually
I just want to give people a hard time, right?
I know, but I'm on board. Like, how easy is this to implement this EMG setup into
It's very
freely behaving mouse?
Yeah.
It's very easy. So look, we did. It sounds presumptuous. We're self-taught in everything electrical because I built stuff in grad school before that. So, but now this guy named Sam Sobel at Emory built electrodes that are, basically he takes PMDS, it's the silane, and will evaporate many layers, and in each layer he'll have an electrode. So the wires are very simple. The problem is that they're if they're too thin, they just jump all over the place.
Yeah.
If they're too thick, they'll mess up the movement. I mean, we found that 25 micron wires are magical. You can put them in the tongue. People say, no, it won't work, it won't work. People say everything without trying it.
And it's like during behavior, like free.
Yeah, during behavior. They'll measure I mean, that's what we did with Sung Ma Lao. We did that behavior paper. We wired up all the muscles in the neck. We didn't publish this. There's a lot of data unpublished. We wired up the tongue. There's no problem with the wires. And the pain in the ass is that when you have many EMG wires, you have to really keep careful track. Like we would thread them through a tube just to keep them clustered, and then inject them into all the different muscles. You just have to be careful to keep them threaded in tubes and stuff to keep them together, and then we would solder them onto a connector, et cetera, et cetera. So Sam, okay, the advantage is, we could put them wherever we wanted.
Can I ask something?
Yeah.
Like how the muscle link is.
It doesn't care. The wires are flexible. They're flexible. They're just flexible. You take the little wire, it's flexible. Yeah, people would say that, but they didn't know what they were talking about because they never tried it. It's flexible. You measure the animal licking in the task, and then you apply the EFGs, and you measure it licking in the task, and the statistics don't change. And I mean, they're not unionized, so they never complain. It's fine. But I thought what Sam did was, a given probe has many contacts, so it's not as good to measuring as many different muscles at the same time, but the business of keeping the wires in order became really trivial. It has a little pad for a connector on it, and it's silicone, it's very floppy, but it doesn't kink or intertwine. So you don't have to do any soldering. It just go. It's just thread it and go. So I mean, I would recommend it. I mean, I wouldn't think twice using wires, but if we're starting from scratch, I think students will get mad at me today if I told them to use wires unless, there's something special. I think for the neck, we tried to get as many muscles as we could in one shot.
And then you still just put like this
pairs.
I
never, the sound things I looked at, I never used yet. I could see what's going on, and I see that people who shied away from wires use. Look, anything we don't have to solder is preferable. Because it's tungsten wire, and to solder it, you have to put on this acid flux. This stuff is easy.
Yeah.
I mean, people like to avoid doing it.
So Sam Sobel where?
Emory, Georgia.
Yeah.
Or maybe it's Georgia Tech, I don't know, kind of brother sister.
Oh, yeah.
Yeah.
Yeah, you have a question?
Yeah, another thing I I wanted to say. We doubt that the IRT also gets projection. We look also at the projections from the SNR to the IRT.
Yeah.
And we want to see how it's related to linking.
Yes.
So we also doubt that the SNR also projects not in here excitatory inputs to the IRT. So we doubt that the IRT gets another excitatory
SNR has excitatory inputs?
to the IRT. We doubt this. This is what we.
I thought they're all inhibitory.
Yeah, so the thing I was talking about that we recently found out there was a problem with the retro viruses targeting both striatum and SNR. But for a while we were using just retroviruses to try to do this, and then we locally injected, and we saw that there's a bunch of CAMK2 cells in the SNR.
Yeah.
And we just got interested in CAMK2 cells in the SNR.
Oh, interesting.
And there's Weber Gear in 2014 has this paper describing this minority population. There's CAMK2 TH+, CAMK2 TH-, and then just TH. And he described all this projection to the thalamus. We got a few cells, with this retro AV. We're now actually wiser, and from the McKelvey paper where we got the lenti viruses to actually try to go downstream of the SNR and target.
But you believe that this is true? There's excitatory cells.
So we don't know if they're excitatory, but the described population that was only described projecting to thalamus were described as being excitatory cells, and they excited a reticular thalamus in this paper, and then no one's really followed up, and we just kind of accidentally fell on this.
What part of thalamus?
I think it's the reticular nucleus, the part that inhibits the rest of the thalamus.
Oh,
that inhibitory sheet.
Reticularis.
Yeah.
Interesting. Okay, interesting.
Yeah. Anyway, so we're just sort of interested in that. But we're mainly, I love to hear your thoughts just generally about this whole angle of trying to understand basal ganglia function by understanding SNR function vis-a-vis these downstream.
So I don't have a good, to be honest, I don't have a good basal ganglion brain. I don't think that way. I don't have any deep insight into the whole system. Lauren, when she came, it was really a means to gain knowledge about a different input pathway into the medulla. It wasn't a means to understand a feedback loop from striatum to substantia nigra to motor thalamus and back. Maybe that'll happen someday, but my head hasn't been there.
Yeah.
I don't have. It is clear because Rui Costa is a co-author on the paper. The truth is, I was actually much more interested, and with things had to do with the nature of the statistics of synaptic connections. There was sort of some arguments over this. I was very interested in the fact that you went to a target and you had a high density of connection, but there just weren't many. And most of them were collaterals along the way. That's what the cells are doing, we're dropping collaterals. And I thought this was interesting. But
the density was very low in the midbrain?
Yeah, cells would project out of SNR. We have a plot, right? And there'd be places where they had a very high density, like boutons per unit area. Right, but then you'd integrate how many boutons there were. And that would be a small fraction of all the boutons. The cell was mostly putting out collaterals. So it had very low density outputs all on its projection pathway.
Mm-hmm.
Which people didn't talk about, which was sort of to our credit, that she was really sort of mapping the density everywhere. So we measured this, and it seemed to be the story for, basically every projection, that you basically were dumping most of your synaptic vesicles in a diffuse manner. So you're telling everybody what you were doing. And so, I mean, it just changed the way you thought about computing, right? It wasn't A to B. It was A with, I mean, somehow you're sending this information to lots of places.
Yeah.
I don't know what to make of it, but I think it's important. Right? It just sticks in my mind as important.
Yeah.
And then it goes to some place where the density is very high. Presumably, it will drive neurons. You know, I don't know if it's in this phase.
Which place?
Where-
The density is very high?
Well, like PPN had very high inputs. You know, some of the thalamic nuclei. I could get the paper. I don't. We kind of. Oops. Did I read this up there? Computers. Hey, Rat, I think you're. If you want to have a deep discussion about the basal ganglia, you should talk to her. Zoom with her.
Yeah.
Right? Yeah, just zoom with her. Just,
And specifically, the basal ganglia is this connection downstream to these.
yeah, okay, so we're like, so. Yeah. Wait, that was her gig. Okay, so we had that, you know, for every region, we had projections to all these different regions, right? So then, yeah, I think this was, at least for me, I had to tell this thing what you want to do, because I'm not here. I want here. Scroll. Yeah, I think this is what I mean. And, okay, so laterals, SCs, pericurriculars, BA, these are motor thalamus, PPN. These are lateral superiorcolliculus. So, you know, this was maybe 1% of all the connections, okay, but they're very dense, right?
Mm-hmm.
The Bhutan density is dense. We pick one. We pick PM as the reference of one. Okay, so places where it's very dense, but you're a much higher, much higher probability of having a low density connection, which is to lots of places. The number of places you had a high density was low. I mean, maybe.
I got you.
Okay, so here's Bhutan density relative again. So we're just looking at the density, and I mean, you're getting almost this log. It's like a log plot of density. Okay,
So basically, in thalamus, you basically have these like very diffuse projections, and then there are these small places where you have very dense projections.
yeah. I think that's what I would say is the essence of it. Yeah, I would say that's the essence of it. And there's lots of places with relatively low density connection. Okay, I'm not such a big. I don't try to play a game with statistics, but I think it's the picture, right? I mean, it seems that the picture in the brain is that you may want to project someplace, but you're sending axons everywhere. I mean, maybe they're ignored for the most part, but maybe during some plastic operation where you're getting some, coincidence, synapses start to form. I mean, that seems to be a basis of behavior. You need kind of a switchboard, right? It doesn't mean that we plug in.
Yeah.
Just need a switchboard.
Yeah.
So, okay, anyway, this is my big contribution to the paper.
No, that's very cool.
Doing
Doing
a MATLAB calculation.
Hassan Grillner was like really, he wrote a preview in this paper being like, yeah, it really changes what is the efference copy for what? Like,
Sorry?
there's the SNR that project the SNR, bifurcation to thalamus, projections of thalamus, and then the projections to these midbrain and hindbrain regions. So like, what's driving the action, and what's kind of like updating, the cortex and the rest of the brain. Like, this is what we're doing, like.
That was our thought. I mean, that sounds extremely qualitative. Julia made a cover, right?
Yeah.
Yeah, wait, I'll show you. Julia made this hilarious cover. It's hard to convince on the neurons to get a cover, but what is it? Sorry. Yeah, Julia she made this. When you're telling the brainstem what you're going to do, and then you're sending messages up to cortex to thalamus saying, I'm doing it.
Yeah, I just think that's
Kind of like-
super cool and influential idea. Like,
Well, it was like me coming here. The day before I left, I told the dean, I'll see you in 2 weeks, but I'll be back exactly 12 hours before I'm officially supposed to send in a request, so don't bother me.
well,
Okay, good.
Yeah, so maybe we'll get in touch.
Another question
Yeah.
about the oscillation and the breathing
Yeah.
stuff. Also, like there are kind of experiments that they do, like they test illusion with whiskers, like by stimulating the VTA and like stimulated regions in the brain, and then they let the mouse think that there's like
Yeah, there was a bunch of pigs. Okay, so there's an argument. There were a bunch of experiments done. It was done in Carell Swoboda's lab, and it was mainly. He's at USC now. Hires, Andrew Hires, Senator Andrew Hires. I think that these animals, they just were not whisking. I think they were in this other mode. So they're just